Let's talk about sampling or testing methods. Two different, I guess, sampling or testing methods. There's a bracketing sampling method, which means if you have a series of testing conditions, you can choose two extreme maybe samples within the range for your testing. And you can assume that intermediate conditions should be okay if both, I guess the minimum and maximum conditions comes out to be okay. Okay, so that we consider as a bracketing testing or bracketing sampling method. Okay, so for example, let's say you have maybe three different I guess the content strength of the pills that you are making, or it doesn't have to be pills, it may be injectable drugs. You're making like a 2 milligram, 4 milligram, 6 milligram quantity of the drugs, samples that you are making, and you're testing those. Then instead of testing all, you can choose maybe minimum quantity, which is 2 milligram, and the maximum quantity of 6 milligram, and do the testing for your stability. Okay, and if the testing comes out to be okay for both, you can automatically assume that in between the intermediate condition, which is 4 milligrams, should be okay. So that's the bracketing testing method. Okay. On the other side, matrixing method is you can select all subset of the total number of samples and test them all. That's the matrixing method. Okay. So there are two different, I guess, testing or sampling methods that are out there. Okay. And then you also need to test for the extractables and possible leachables. You probably heard those two terms a lot, right? Extractable and leachables. What's the difference? Okay, so extractables are part of leachables, meaning some chemicals can come out from maybe the containers or whatever maybe the manufacturing vessels or things that are making contact with your drug. Okay, so that means like a leachable if they come out passively very slowly, okay, and mixed up with your drug, okay. But if they are coming out kind of under the artificial condition, under kind of a vigorous exposure, we consider them as extractables. So extractables are kind of a part of the leachables as well. Okay, so anyway, so extensive extractable studies should be performed as part of the qualification of the container closure components, including labels, adhesives, and inks. Okay, so make sure that there's nothing that's coming out from those and mixed up with the drug, right? And then absorption or adsorption. Okay, absorption or adsorption of a drug product must be also evaluated during the stability of the studies. Again, then what's the difference? Absorption versus adsorption. Okay, it's hard to pronounce. So the difference is what? Adsorption is adsorption is if your drug is sticking to the surface of whatever the things it is making contact with, maybe the primary container, vial, or syringe. We consider it as adsorption, okay? But absorption is not only it's making contact, maybe it's going into the material, maybe it's the syringe or vial. It's going in, penetrating through, and that we consider as absorption. It's absorbed through. That's what it means. So neither of them should happen. Then you'll lose your drug, of course, from the content, right? So you probably don't want that. Okay, so that should be evaluated, so you don't lose your drug from the packaging material containers. And the leachables are potentially problematic, of course, with drug stored in the plastic syringes, as leachables can occur during from glass and rubber closure system as well. So stability studies must evaluate the integrity of the tip cap, needle, stopper, syringe, and etc., all those things in the temperature and pressure, different conditions. For products containing antimicrobial preservatives, acceptance criteria for such chemical content should be established during the product shelf life. Okay, we talked about PAT test right before, so understand that. So microbial challenge test, that's a PAT test, should be conducted using a preservative level less than minimum amount specified as acceptable. Okay. And the sterility, it's not stability, it's the sterility must be monitored. Okay, sterility means what? There's no microbial coming in and contaminating your drug. That's what sterility is. must be monitored throughout the stability testing period. Okay, so it's kind of confusing, stability versus stability. So stability determines your shelf life, right? During the shelf life, your drug should be remaining sterile. That's what it means. Okay, there's no contamination happening. So, test on the integrity of the container closure system must be conducted annually to make sure that your drug is still sterile. And pyrogen, bacterial endotoxin testing is also needed. Okay, so FDA stability guideline. Okay, solution dosage form, stability data should be generated for the active ingredient. Okay, how much is also, until good during the shelf life time, the dosage and I guess activity and everything, and the antimicrobial preservative if present, so PAT test should be done, of course, and then appearance and any change in color and clarity should be also tested. And degradation of a product, of course, important. Particulate matter, there should not be anything that's coming and becoming particulate matter in your drug. pH level should not change, sterility should not change, and pyrogenicity should not change. Okay. And the sterile products that will be reconstituted into solution, freeze-dried drugs, immediately after reconstitution, stability data should be generated for, okay, like we talked about 24 hours, if it's not refrigerated. If it's refrigerated, 48 hours, they should be still good, right? So not only that, you need to test all these things. Residual moisture content right before reconstitution. Okay, so you're going to do the lab on this residual moisture content using Carl Fisher titrator. Okay, there's a way to measure that. And then the active ingredient of the drug, okay, how good it is. And then appearance and any change in color and clarity, of course, same. And the particulate matter, reconstitution time, less than 2 minutes. And then pH, no change, sterility, no change, pyrogenicity. Okay. At various time after reconstitution, during the 24 hours or 48 hours time period, the active ingredient, if it's still there, okay, without the degradation, appearance doesn't change, pH doesn't change, particulate matter should not be there. Okay, all these things should be tested. That's what FDA wants to see. And if it's a dispersed system product, if it's a suspension, for example, particle size distribution should be measured, and redispersibility, should be measured. Meaning, when you shake it up, how long it stays there as a suspension. That's what it means. And physiological properties like flow and deformation under pressure. It's hard to say those terms. Okay, should be also tested as well. If it's an emergent drug, okay, mean globule size and distribution. Since it's a lipid droplet that's going to be in your drug, what's the size of those and how well they're distributed should be measured and tested. And the phase separation. Okay, if lipid phase or water phase lipid phase, they should be measured, how well they're separated, how they'll mix when they are mixed up, and how long they are staying as a mixed form should be measured. Viscosity, if it's a heavy form, okay, how viscous it is should be also tested. Products contained in pre-filled syringe, if it's a syringe-filled drug, then applied extrusion, that's brake force, okay, how much power is required to move the plunger. That's what applied extrusion breakpoint is. And then glide force, movement of the product through the barrel of the syringe, okay, should be also measured. And syringeability, how easily can you inject the original syringe. And leakage should be, of course, checked. And if it's a combination product, your drug is mixed up with some kind of device sold together, and product to be combined with a diluent example, or other drug product, or comparability, and comparability must be determined in various orientations immediately after the combination. If it's a combination drug, two drugs are combined together before it's injected, yeah, they should be tested together, and the comparability should be tested after longer storage time. Okay. The evaluation should include the data on the test performed on other sterile product as well, if whatever the number of drugs are mixed up together. Okay. Stability data requirements during drug development phases. Okay, phase 1, a brief description of the stability study and test method used to monitor the stability of the drug product during phase 1 clinical trials. Brief description is needed. And phase 2, provides sufficient information to develop the final formulation and the most appropriate container and closure system by the end of phase 2. And clinical trial phase 3, testing on final formulation in the proposed market packaging and manufacture site. A final stability protocol should be well defined prior to phase 3. Okay? And then, of course, everything worked out and you apply NDA, of course. And then after the approval is given, then extra stability studies may be needed for reporting changes for the manufacturing processes. So after the NDA approval is given, I mean, if ideal, nothing should be changed. But if there's something needs to be changed, extra studies should be done on that change. That's what it means. Manufacturing site formulation, addition of new strength for drug product, equipment, batch size, reprocessing steps, container closure, stability protocol. So any change you make, they should be tested. That's what it means. So, the ideal situation is everything is tested before your NDA submission. So it should be the part of the approval, and nothing should be changed. Everything is locked on. That's what ideal condition will be. And distribution and storage. Okay, once unit dosage forms are filled into primary packaging, stoppered and sealed, inspected, labeled, and placed in appropriate secondary packaging, issues of distribution and storage come into play. So that's the final thing that's going to happen during the drug product manufacturing. So good practices for storage and handling of parenteral drug products are very important, of course. So you need to keep them, if it's refrigerated, constant temperature. No fluctuation. Excursion means fluctuation of temperature should not happen. Outdated shelf life, so within the shelf life, they should be kept, of course. And glass hairline crack that cannot be easily detected are common problems. So during the maybe packaging or during the transportation, some damage could happen on your vials and syringes. Okay, that should not happen. That's what it means. The shipping process must be validated to assure product stability, of course. Temperature must be controlled. The packaging system must have protective propriety such as light protection, insulation, cold storage compatibilities. Cold chain distribution systems are needed for biopharmaceutical products. Of course, you know that Moderna vaccine needs like a minus 80 degree freezing condition. So you have to have that cold chain distribution system in place for your drug. The effect of unintended freeze-thaw cycle on products stability must be studied. What if there's electric outage, like things that's happened in Texas? Then what's going to happen to your drug? Should you throw everything away? That's going to be a big waste. So that's what I mean. All those things should be studied. So you have something to say if something like that happens. Okay. So that's the end. So finally, the assignment question. So according to USP, US Pharmacopoeia, what are the particular matter in the injectable or parenteral drugs? That's undissolved matter into your drug, excluding gases, right? That's what the USP says. So right there. Okay. Particular matter in injection and parenteral infusions consist of mobile, undissolved particles other than gas bubbles. They are unintentionally present in your solution. That's what particular matters are. Okay. And then, how do they affect patients if the drug containing them are injected into a patient? What's going to happen? They may have embolism, that's a blockage of the blood vessel, or huge inflammation, right? So where do we talk about? Granulomata, that's a huge inflammation, pulmonary granulomata, it's in your lung, which is not good, and blockage of the blood vessels, emboli or embolism, that may happen. Okay. So list 3 of the tests used in package integrity testing. We talked about 7 different testing, just pick 3 and list them. Okay. And what are the 2 main requirements with respect to stability studies stated in the GMP guidelines? So stability studies, what are those? Where can you find them? Okay, so here are requirements to a written testing program designed to assess the stability characteristics of a drug product, which is used to determine appropriate storage condition and expiration date. Okay, and then another one is requirement for an adequate number of batches. Okay, should be tested. Okay, so those are the two. Now, what are the storage condition requirements different for drug products